Hello and welcome to this segment on cloning and genetics. Here we will talk a bit about cloning and genetics using somatic cells. To start, let's go over some definitions. Genetics is an evolutionary field of science that deals with the DNA in cells and genes found in organisms such as animals, plants, bacteria and fish. Cloning is a set of process that produces an identical organism. There are various ways to clone an organism, one of which is using somatic cells. Somatic cells are any cell in an organism with the exception of reproduction cells. For example, cells found in the lungs, liver, skin, brain tissue, pancreas, blood, saliva, arteries, veins, toenails, and hair follicles. Somatic cells are not oocytes, eggs, or sperm. Cloning an organism requires 3 donors. 1 somatic cell donor, 1 oocyte donor, and 1 surrogate donor. The process of cloning using somatic cells is divided into 6 basic steps. Now that the definitions have been covered, it is time to move on to the 6 basic steps in cloning. Keep in mind this is just a basis which does not actually include every single step, as there are many processes. To begin, know that there are 2 ways to clone, but for now we are using the somatic cell nuclear transfer process. The 6 basic steps consist of obtaining a somatic cell from donor, obtaining an oocyte or egg cell from donor, extracting the nucleus from the oocyte, inserting somatic cell into oocyte, giving an electric pulse stimulation to cell to start the division of cell process, implanting the hybrid cell, otherwise known as the embryonic stem cell, into surrogate mother donor. Next, you will notice some pictures. The first picture is of a donor oocyte having the nucleus extracted. The second picture is of an oocyte having the embryonic stem cell inserted into the donor oocyte. The third picture represents the cloned animals after the process has completed. Next is a short video that further explains the process of cloning using somatic cells.
This video is prepared by my colleagues Dieter Egli and Kevin Egan. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out. And then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus, and if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore, and there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you 2, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again, a little hole, and the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer.